Our skin is a complex organ that contains a highly specialized immunologic system that is used for the maintenance of tissue homeostasis, defense, and repair. To discuss the structure of the skin and its intrinsic defensive mechanisms, we have the pleasure of welcoming Dr. Georg Stingl on the podcast, who is a world expert on immunology of the skin. In other words, an immunodermatologist. Dr. Stingl is the chairman of the Division of Immunology, Allergy and Infectious Disease, and the Department of Dermatology at the Medical University of Vienna in Austria. He has received several prestigious awards, such as the William Montagna Award and the Stephen Rothman Award of the Society for Investigative Dermatology and the Lifetime Achievement Award of the American Skin Association. He is an honorary member of the Society for Investigative Dermatology and the European Society for Dermatological Research, a member of the Austrian and German Academy of Sciences, and in 2013 was elected foreign associate member of the Institute of Medicine in the U.S. National Academies. Dr. Stingl, it is a real pleasure to have you on the Derm Club podcast today.
Thank you very much for the very kind introduction. And you have mentioned everything that is of importance. There's only one thing I would add to define my role as a physician and as a scientist. Yes, it is true. I am an immunodermatologist. In other words, I look at patients with immunologically mediated skin diseases, but at the same time, I'm also kind of a cutaneous immunobiologist who did and does kind of basic immunologic research with the skin as the major substrate.
Okay, thank you for adding that. So there are many different layers to the skin. Then from an epidemiologic standpoint, why do we need all these layers? You know, we have the epidermis, we have the dermis, and we have the subcutaneous fat.
So in general, we have 3 layers that you just mentioned, and for quite a long time, we defined the skin simply as a physical barrier organ. And in the meantime, we know that this definition is not enough. There's no question. The physical barrier is very, very important. And in this regard, the 3rd layer, the subcutaneous fat and the adipose tissue, is a very important cushion that protects us against too heavy of a trauma. But this is certainly not enough. In the meantime, we know that the skin is a physical barrier. The skin is a chemical barrier. The skin is an immunological barrier, and the skin is also a microbiological barrier. And God knows what we will find in the years to come. So it's a very multifaceted understanding of the protective role of the skin. The challenge for the skin is pretty high. It has to be, we have to separate ourselves from the environment to some extent. In other words, we should not allow that pathogens or some kind of dangerous subjects, objects, or let me just simply say danger signals reach us. But at the same time, we cannot completely separate ourselves from the environment. So the skin, especially the epidermis, has to be like a sieve, like a tea sieve, where we are able to exchange fluids, sweats, but it should not look like a Swiss cheese. The holes should not be so big that bacteria or large proteins and whatever enter the skin. And if this happens, then we have barrier defects and skin diseases caused by these barrier defects, such as ichthyosis, such as atopic dermatitis, and many others.
So how do we maintain that balance? so that we don't have too much, but not too little?
Okay, so thank God we have, under normal circumstances, a pretty good genetic equipment. This is very, very important, that the regulation of protein expression and gene expression in the skin runs in a correct fashion. And we all know what happens if protein synthesis, if the regulation of protein synthesis does not work, then we have big troubles. If you look at patients with ichthyosis, for instance, who have mutations in the filaggrin gene, you are all familiar with children with epidermolysis bullosa who don't have an appropriate attachment between the epidermis and the dermis. So what I'm saying is the basic requirement is an appropriate function of gene expression in the skin. This is a prerequisite. But number two, there are many epigenetic factors that influence the correct functioning of the skin. I think best known to most of the people is ultraviolet radiation. Ultraviolet radiation can lead to a disturbance not only of the physical barrier, but mostly of the immunological barrier. If you have photoaging or UV irradiation or unprotected UV irradiation for too long a time, then we know very well that the immune function of the skin is no longer maintained. And this causes, together with direct carcinogenesis and keratinocytes, this also causes immunosuppression. And this type of immunosuppression makes it more likely that patients or people will develop skin cancer, white skin cancer, but also pigmented lesions and others. So it is a malfunctioning of both of the genetic basis and the epigenetic influences that favor the development of skin pathologies in general.
So I know you touched upon how skin really acts as a barrier to avoid any infecting pathogens that are trying to get in. Can you give us some more details about that, and which things play an important role in protecting us from infecting pathogens?
Okay. There is some knowledge that we gathered from observations of human skin and human disease. And there are other observations or other concepts and hypotheses developed on the basis of experiments with or studies in experimental animals. And in the case of the immune defense, we have data both from human studies and from animals. There is one discovery that is rather new. And this was derived from studies in mice. And we now know that commensal bacteria, totally innocuous, innocent bacteria that we all have on our skin, Staphylococcus hominis, epidermidis, and many other of the so-called commensal flora. These bacteria, they are not innocent bystanders. What they do, and we don't see that, this runs subclinically. These commensal bacteria, they activate the immune system, predominantly the adaptive immune system. This works in a loop that goes via dendritic cells and T cells. So in other words, these commensal bacteria, they train our immune system, the adaptive immune system of the skin, in such a way that the skin will be better prepared if a true challenge with pathogenic bacteria occurs, such as with Staphylococcus aureus or Pseudomonas and several others. So, so we have kind of a subclinical immunological defense system. And what happens if we have a major microbiologic challenge? Then perhaps this system will not suffice. The system that I have just described will probably not suffice to protect us sufficiently. In the case of a major microbiologic challenge, we get the activation of both the innate immune system, or elements of the innate immune system, and of the adaptive immune system. We all know that the adaptive immune response is a very, very rapid one, but it's also not very specific. It is kind of a promiscuous response, happens almost immediately after the occurrence of the challenge. And there are certain cells and certain molecules that are a major part of this innate immune response. I mean, we all know neutrophils. We all know monocytes and macrophages. We also know molecules such as antimicrobial peptides that have the capacity to kill bacteria. And if patients with atopic dermatitis, for instance, cannot produce these antimicrobial peptides in sufficient amounts, then they have problems. They have problems with staphylococcal superinfection. There's also a new population of lymphocytes that belong to the innate immune system that we recognize only since a few years. And these lymphocytes are called innate lymphoid cells. They don't have antigen receptors, but what they do have, they have receptors where they recognize patterns on microorganisms, patterns on damaged cells, DAMPs and PAMPs, and then they become activated. And what these elements of the innate immune system do is they attack the pathogen right away, but at the same time, they also prepare the elements of the adaptive immune system to be better responsive to the microbial challenge. And it is somewhat surprising, but we know the various cellular and molecular constituents of the adaptive immune system for a longer period of time than the innate immune response, even though the innate immune response comes first. But in the last 50 or 60 years, when we learned a lot about skin immune functions, we focused at the beginning predominantly on the adaptive immune system. We realized the importance of dendritic cells, Langerhans cells in the epidermis, dermal dendritic cells in the dermis. We also know now that there are also dendritic cells in the fat. And then we characterized the T cells of the skin in a very detailed fashion. And we know a lot about the T cells. We have also learned that certain lymphocytes don't like the skin. B cells don't like to go to the skin. And K cells, there are also not many NK cells in skin. But T cell function and activation of T cells by dendritic cells, this is very well investigated. And so if you wish, there is a two-step process: challenge and threat by a danger signal. Step number one, activation of the innate immune system. If successful, this eliminates the pathogen right away. If the innate defense does not suffice, there is activation of the adaptive immune system. As I just said, the innate system helps to make the adaptive immune system well tuned to these danger signals.
So in diseases like atopic dermatitis, psoriasis, say, is our thinking now that the innate immune system is really not cranking up and working efficiently?
This is not a black and white answer that I can give you. In the diseases that you mentioned, in atopic dermatitis and in psoriasis, we have very, very good evidence that both the elements of the innate immune system and the adaptive immune system play a very important role in the pathogenesis. In the case of atopic dermatitis, we have lots and lots of T lymphocytes. These lymphocytes are mostly of the so-called type 2. In other words, they produce predominantly cytokines such as interleukin-4 and in humans mainly interleukin-13 and also interleukin-5. But in the meantime, we also know that in addition to the T cells, we also have innate lymphoid cells in the skin that produce the very same cytokines or very similar cytokines. What we do not yet know is what is the relative contribution of the adaptive response and the relative contribution of the innate response. This is not a trivial question, because if we try to therapeutically influence the skin immune system, it makes a difference whether we try to target predominantly cells of the innate response or of the adaptive immune response. But this is being investigated in great detail by several groups around the world, and we have learned a lot in the last 3 or 4 years, and I guess we're going to learn even more in the years to come about this particular question.
What role do you think the microbiome plays?
The microbiome, first I should say, are we talking, is your question directed to the cutaneous microbiome or to the microbiome in general? Probably the cutaneous, right? Or both, right?
Yeah.
So, so in the case of the microbiome in general, I think most of the evidence that we have and most of the studies that have been done were dealing with the intestinal microbiome, and you and I and we all are familiar about possible connections between malfunctioning of the gut microbiome and certain diseases, like even metabolic diseases. People maintain that diabetes and cardiovascular disease and many others are in some connection to the functioning or functioning of the microbiome. We also know, and there's a lot of speculation about this, that there are gender sex differences in the composition of the microbiome between men and women, and it's also not entirely clear whether these difference in the microbiome between men and women is perhaps responsible or co-responsible for the fact that certain diseases are more frequent in men and other diseases are more frequent in women. That's possible. In the case of the cutaneous microbiome, I should say that we have several layers of the cutaneous microbiome. We have, I just mentioned already, the commensals that are out there on the surface of the skin, but we also have an indigenous microbiome at deeper layers. We know most about the role of the epicutaneous and the epidermal microbiome, and one of the roles of the epidermal microbiome I have mentioned already, and this is the fact that some of the bacteria that we have, some of the innocuous bacteria that we have on top of our skin, that they induce a subclinical, non-visible immune reaction that makes our immune system fit for the challenge that we have in the case of a real pathogen. This is probably very important, also because of the fact that the bacteria that we know that are on top of the skin, many of these harmless staphylococci and also others, they can produce antimicrobial peptides, and they can kill Staph aureus. This is probably one of the reasons why, especially in patients with atopic dermatitis, where we have major problems with staphylococcal skin infection, people like the group of Dr. Gallo in San Diego and others, they have tried to make transplants of a functioning cutaneous microbiome from a healthy person to individuals who suffer from very severe atopic dermatitis. I mean, these studies have not been completed, but it's not yet clear whether this approach will work. But I think what we always should keep in mind, as dermatologists, we often said when patients had skin super infections, right? What we often did, we either used topical antiseptics, we used bleaching, we administered antibiotics to our patients, and only focusing on the presence of Staphylococcus aureus. And we often did not think about the fact that we also destroy or damage the good part of our microbiome. And this is an issue or a question, a problem that we have not yet resolved. There is no question that if you have severe infection of the skin that you will use antibiotics. That's not the point. But we should not be too easygoing. If you see a few follicles and you see a few pimples, that we immediately start with systemic antibiotics. We have to be a little bit more careful and allow the components of our skin microbiome to defend ourselves.
How do you feel the same way about steroids?
Yes, to some extent. Yes, to some extent. But I think in the case of corticosteroids, I should make another statement, which I think where many dermatologists make major mistakes. Somehow, especially in the US and Europe, and also I guess also in Japan and Asia, there is a certain corticophobia in the population. So kind of as if corticosteroids would be the worst in the world. And this is not true. Sometimes corticosteroids can be very helpful. But we as physicians and also as dermatologists, we often give wrong advice. In the case of corticosteroids, I think the most important thing is to administer them in a workable, robust concentration, but for a short period of time. So you should allow them to work. You should allow corticosteroids to interrupt a pathogenic loop, but then you should try everything to lower the concentration and then to stop it. The mistake that we often do is we use extremely weak corticosteroids. We apply them to the skin. We tell the patient, you apply these corticosteroids to the skin. They do it for 1 week, 2 weeks. The whole thing doesn't get better. So they continue to apply it further. And the result is the clinical improvement of the skin disease does not occur. But at the same time, if you apply them for 3 or 4 weeks and longer and longer and longer, you get skin atrophy, telangiectasis, and all the other things. So I think we as physicians should advise our patients in the correct fashion. And then corticosteroids are an important part of our therapeutic armamentarium.
So you mentioned using mice models. Why do we choose to use murine mice models? Are there limitations, and are there any better models to mimic skin immunology?
That I really, the last person who denies the importance of animal models in general, or mice models. And in my own research, I have worked a lot with mice, and but we should also understand that some of the investigations that had to be done with mice, simply for ethical reasons, but also for technological reasons, could not be done with human tissues, and there were certain limitations. These limitations, to some extent, still exist. But what I'd like to emphasize is we have now new technologies where we can investigate the human substrate in great detail, and we can extricate important information. I guess you and all the others, we are familiar now with single-cell RNA sequencing. We can look at individual cells in normal skin and diseased skin, and can get very important information as what they do and how they function. So the issue is not to completely replace animal models. Certainly not. The animal models will always have their place because certain things cannot be done in man. But what I'm saying is some very important information about disease pathogenesis and also about therapeutic targets can be gathered by human investigations. And I think this is to me, is the great excitement that we can do that now, that we can take 4 millimeter or 6 millimeter biopsies and can get an idea about the whole repertoire of the different skin cell populations. I think it's an exciting time. And we should also not forget, we still don't have the ideal models for psoriasis. We don't have an ideal model for atopic dermatitis, not even for contact dermatitis. And actually, if I take away certain genetic diseases, I know of only very few models that exactly mirror breaking human research. We should use that in order to make progress.
Amazing. It shows where science is really going,
Right.
and how far it's come.
Yeah, and also what goes together with it. I mean, we now have this revolution in analysis and interpretation of data, the area of artificial intelligence that influences our type of research. We can gather important information from all these sequencing data, for instance. And now we also have the technology to interpret this data and display them. This is an amazing development, mostly in the last 10 years. I think it's fascinating. You have, in your young age, you have great decades of excitement in front of you.
Thanks to you, because you've opened those doors. So
A little bit.
we didn't really get to talk about Langerhans cells. Can you please share with us the importance of Langerhans cells, what they do, how our understanding of Langerhans cells have really changed how we treat chronic skin disease?
I want to make that short, and I want to divide my answer in three portions. Portion number one, there was a medical student in Berlin in 1868, I think. And he was a very gifted guy. He discovered two things. He discovered cells in the skin, where he thought these cells are part of the nervous system. And then, also known to every physician, he discovered the islets and the pancreas, right? And the cells in the skin, for 100 years, we had no idea whatsoever what these cells are doing. And many people, actually, some very famous dermatologists and skin researchers, thought that these cells are an artifact and that they don't even exist or just are dead. A very frequent theory was that Langerhans cells are dead melanocytes, right? Mm-hmm. And then we and others in the 1970s, we discovered that these cells are not dead, and we found out that these cells have typical features and typical phenotypic features of cells of the immune system. And very much at the same time, investigators in New York, Dr. Steinman and others, discovered dendritic cells in the spleen, and the Langerhans cells looked also dendritic. So the idea came up, there is perhaps in the whole body a system of dendritic cells that is of immunological importance. We and others then found that these cells can stimulate T cells and are very potent stimulators of T cells. And the idea came up that the immune responses in the skin, that they all originate from Langerhans cells. The Langerhans cells that sit outside in the epidermis. So if some kind of dangerous bacterium comes along, they can pick it up and activate T cells. This was a very simple view of the function of these Langerhans cells. Also because people found out that Langerhans cells are not the only dendritic cells in skin. Langerhans cells are the major dendritic cells in the epidermis, but there are other similar dendritic cells in the dermis. And then it turned out that under homeostatic conditions, if there's no challenge, Langerhans cells are probably important to prevent an activation of the immune system. In other words, it wouldn't make sense that if we would mount a very potent immune reaction against innocuous birch poles, it would also not make too much sense to mount a productive immune response against human dander if we scratch our, it doesn't make sense.
Right.
We should rather prevent the occurrence of such an undesired response. But on the other hand, if there is a major challenge, a major pathogenic challenge, then these cells should be prepared to do that. And this is what our understanding currently is, that under homeostatic conditions, we should rather prevent the occurrence of exaggerated immune responses. But in the case of real danger, Langerhans cells, but perhaps even more so dermal dendritic cells and others, they should mount a strong response. This is our current understanding. But we should also not forget Langerhans cells in the epidermis. They are in a very intimate, extremely intimate contact with nerve endings. We know that, but we don't know yet what that means. In other words, what I try to say, even as someone who has contributed to the discovery of Langerhans cell immune functions, I think we should not exclude the possibility that Langerhans cells might have functions that we do not know at the moment, and maybe may have nothing to do with immunology. I think it will be very interesting to find out how the interaction between sensory nerve endings and Langerhans cells does. How does this influence the nerves? How does it influence the dendritic cells? And many, many other really unresolved questions. So it's still worthwhile for a young investigator like yourself to say, okay, I'm going to embark on this and I'm going to do intelligent experiments.
Maybe I'll come to Vienna and we'll embark on it together. So we mentioned that when you have a dysfunction in the normal homeostasis of the skin, you see diseases like atopic dermatitis, psoriasis. What other diseases do you see when you have this dysfunction of normal homeostasis?
Well, I guess a dysfunction of normal homeostasis can be caused by immunologic mechanisms, but also by totally different pathogenic occurrences. Obviously, in the case of immunology, I think we should not forget this whole large group of connective tissue diseases that, in my view, they don't get enough attention from us dermatologists. I think we as dermatologists, we should be prepared and able to treat patients with lupus, with dermatomyositis, with scleroderma, because if we are not careful and attentive, there are rheumatologists and others who might take kind of these diseases away from us. So we should pay attention to these diseases. I'm also very interested in this large group of neutrophilic diseases. In the case of psoriasis now, but whenever you go to a meeting right now, you constantly hear about psoriasis, atopic dermatitis, lymphocyte-mediated diseases, lichen planus and others. But I think I'm also quite fascinated by the occurrence and by the mechanisms that drive neutrophilic diseases like acne. We don't have a full understanding of acne yet. Is there an adaptive component? Yes or no? Can we, by investigating the type of lymphocytes, but also the type of granulocytes present in the lesion, can we actually open totally new avenues for the treatment? A disease which, thank God, has received a lot of attention, or is receiving a lot of attention right now, is HS. It's a terrible disease, hidradenitis suppurativa, and we have learned a lot. We have learned that obviously Th17 cells are a major player. So we are developing now new concepts for the treatment of those types of diseases. I think we also need more innovative treatments for diseases like pyoderma gangrenosum. What we still do is the patients receive corticosteroids, the patients receive cyclosporin, and perhaps TNF inhibitors. But I think by thorough investigation, we can do much more. And so there's a whole field that has to be discovered, and in general, I think our attempts, our therapeutic attempts, and our research attempts can be summarized in one sentence. And the sentence is, in the search of specificity. When you look at the development of drugs, right? And you go back to the, let's say, 1950s, 60s, we had corticosteroids, we irradiated the skin. Later we had antihistamines, but there was not much. And all these drugs that we have, also methotrexate, they had many, many effects and many side effects. As I said at the beginning, a rather promiscuous approach to treatment. And what we did now, and we only owe this to the support of research. Without science, this would have not happened. We now try to develop more and more specific treatments. And that future and that promise is fantastic. I think your generation, you will be in a situation, I guess, in 10 years, 15 years, we will no longer treat patients with pemphigus vulgaris only with corticosteroids plus Immuran. Even rituximab may not be enough. In 15 years, you're going to say, what I'm going to do is I'm going to eliminate specifically antibodies against desmoglein 3 or desmoglein 1. I leave the rest of the immune system completely unaltered in the search of specificity.
The targeted therapy.
Yeah, a targeted therapy, and the target is getting narrower and narrower and better defined over the years.
You know, just to add to this, we were talking about neutrophilic diseases. I recently just published a case report on patients who get a rash called prurigo pigmentosa. It's a dermatosis, and they believe that it's often associated with the
Say it again. I didn't hear it well.
Prurigo pigmentosa.
Okay.
They believe that it's associated with a ketogenic diet. And the idea behind this is that ketone bodies might actually induce perivascular neutrophilic inflammation. And it makes me think, like, what are we using to treat it? Either we could stop the diet, and often dermatologists are also prescribing doxycycline or minocycline because I think that it inhibits neutrophilic chemotaxis and function. But I always find myself thinking, how about if we said we don't need this antibiotic? Let's just eliminate the need for the antibiotic so we don't create more resistance, and let's just take the patient off the diet. And I know some dermatologists are thinking that way, but some are, I think, want to cover both bases, so they prescribe the antibiotic in addition to telling them to return to a normal diet. So it just makes me think about targeted therapies and what you were saying about neutrophilic diseases, and I find it very fascinating.
Absolutely, and I guess, and you will probably confirm that, especially in the case of drugs like doxycycline and also minocycline, many people, or many dermatologists, don't understand that we are using these compounds, especially in diseases like rosacea and others. We don't use them because they have an antibiotic effect. We use them because they also have an anti-inflammatory effect. So the real challenge is we need drugs that we need a doxycycline that by itself is not an antibiotic, but only has the anti-inflammatory effect. Then I think this would be a major progress in our attempts to treat diseases like perioral dermatitis, rosacea, and several others.
I think the closest thing we've had at this moment maybe is tacrolimus.
Yeah.
So what do you perceive as some of the challenges still ahead for immunology and dermatology? I know this is a loaded question.
No, I think the question from an immunologic viewpoint, they will remain what they are. And I think in our discussion we had mentioned several ways how we can pursue them. And the most important thing I see, and this is a shift of paradigms. In the past, we understood ourselves as dermatologists or as physicians in general, we understood ourselves as therapeutic physicians. Mm-hmm. Now, I think we know a lot about skin physiology, and we understand skin physiology not only at a cellular level, but also at a molecular level. So I think a great challenge for us is the maintenance of skin homeostasis. We understand what we need in order to maintain homeostasis. And this is a major challenge from a preventive viewpoint. Number two, the big challenges, the the scourges, if someone is the scourges of mankind, they will remain, right? We have also as dermatologists and as a physician, we have to be prepared to recognize, to diagnose, and successfully treat infectious diseases, allergic diseases, cancer, metabolic diseases. So these changes, they will remain the same. And we have discussed possibilities, what we can do and how we can be more specific in our attempts. Politically, as a dermatologist, I think if we attempt the dermatology in the crown of the different medical specialties, the dermatology maintains its position, we also have to do something. I think in our training programs at the universities, at the hospitals, in medical schools, I think we should really try very hard to train competent physician scientists. If we don't do that, there could be a possibility that dermatology becomes marginalized. And I don't want that to happen. I think we should be the ones who not only diagnose skin cancer, but also treat it successfully. We have to understand principles of allergic diseases and autoimmune diseases in such a way that these areas will not be taken away from us by so-called clinical immunologists and allergy specialists, rheumatologists. These are all things that in our training programs at the university and the hospitals, that has to be considered. Otherwise, we end up as pimple squeezers, and we don't want to, and that's not what's really our goal, right?
Thank you, Dr. Stingl, for sharing this fascinating conversation with us today and for really teaching us not everything, but the basics of immunology. Is there anything you want to add before we close today?
No, I think I made scientific statements and defined goals that we have to do as physicians and scientists, and I also defined political goals as far as the whole discipline of dermatovenerology is concerned. But I greatly enjoyed talking to you, and I thank you very much for the kind invitation. Perhaps at some time in the future, we'll have the possibility to chat again. Thank you very much.
Hopefully in person. I hope you enjoyed this episode of the Derm Club podcast. If you found the discussion today to be valuable, please subscribe and share. I look forward to seeing you in the next episode as we continue to delve into dermatology and skincare with the world experts.